Hello student, good morning. Today we will start the polymorphism in coelenterates. Polymorphism in coelenterates. What is meant by polymorphism? When a single species of coelenterates animals represent by them one kinds of individuals differing in form and functions, it is said to be polymorphic and the phenomenon is called as polymorphism. The heterozone coelenterates, particularly of the order Siphonophora, exhibits polymorphism of highest degree. There are two basic forms in the coelenterata animals, the polyps and the medusa. Pneumatophores, Nyctocalyces, ectoderm, endoderm, entire cavity, cynosphere, heterophilium, dactylozoids, basal tentacles of dactylozoids, gastrozoids, battery of nematocytes. Polyps is generally sessile tubular organisms attached to some substratum. By its base, it bears mouth and tentacles at its free end. It is also feeding zooids or gastrozoids. The medusa can be compared to the polyp in its basic constructions, but it is a free swimming discoidal or bell-like form having considerable development of contractile muscles processes, giving a high muscular cavity activity. The mesogloia is also much developed. The gastrovascular cavity becomes more complex. Besides efficient nerve the medusa shows development of sense organs like statocyst, photo and chemical receptors. The tentacles are generally borne on the margin of to be the bell and not around as in the polyp. Both these forms undergo considerable modifications in the colonial Siphonophora, and at time both the polyphoid and medusoid forms are retained in one and the same colony. Polymorphism explains highest degree of division of labor in a colony. The polyphoids are of three types: gastrozoids, dactylozoids, and gonozoids. Gastrozoids. Gastrozoids are the nutritive zoids of the colony. They are also called as siphons. The polyphoid zoids has a distinguishable mouth and tubular or circular body. At the base of the gastrozoids, usually there is a long and hollow contractile tentacles. It either bears batteries of nematocysts or lateral branches, each ending in a knob or coil of nematocyst. The endodermal layer is generally pigmented and produced into villi projecting in the coelenteron. These gastrozoids can capture large size prey and ingest it. Gastrozoids mouth tentacles tentilla merocyst knob. Lake mouth. Dactylozoids. These are the protective polyps in the colony. These are also called pillars of palpons or testers. They are like gastrozoids but lack mouth. The basal tentacles is never branched and has high concentration of nematocysts in Vetella and Porpita. Dactylozoids near the margins are long, hollow, thread-like. In the Hycelia, they become very long filaments, sometimes tentaculous coelates. Dactylozoids present near the gonophores becomes gonopalpos. Gonozoids. Third, these are the reproductive zoids of the colony or the blastocyst. These are generally with mouth and produce medusoids by asexual budding. In Velilla and Porpita, the gonozoids bears mouth is gastrozoids. In Phacelia, the gonozoids is in the firm branch stalk called gonodendron, which bears clusters of gonopores and often provided with the gonopalpon. Modification of the medusoids. The medusoids are modified as the nematophores or the floats, the swimming bells, the bracts, and the gonophores. Nematophores. It is also called the floats of assumes the form of a vesicle or bladder filled with gas. It established in the entire colony of the float near the surface. Swimming bells or nectocalyces. These are also called as nectozoids. They are without mouth, manubrium, tentacles, and sense organs, but have velum, bell, and 4 radial canals and circular canals. Third, bracts or phylozoids. The medusoids assumes various shapes like a leaf, shield, helmet, etc. Gonopores. These reproductive zoids may be solitary or in the clusters on stalk on polypoid zoids. Example, velilla. They may occur on the gonodendron. Example, X. Phacelia. They are usually hermaphrodite in nature but dioecious gonopores. Hermaphrodite in nature but dioecious gonopores also occurs. These gonopores may retain some of the medusoid characters, particularly the female zoids. The male zoids may be reduced the sac. These pairs may be set free, but when retained in the colony, they die after the discharging the gametes. Ovary, testes, concentric, chitinous, ear, chamber, liver, caul or crest, gastrovascular canal, tracheal canals, gonozoids, edge of disc, gonozoids or blastostyle, large central medusae, gastrozoids, tentaculozoids, dactylozoids. Polymorphism in the coelenterate. This is the complete polymorphism of coelenterate. Polymorphism in coelenterates. Okay, thank you.